Next up is Katy Hayden, who she's Quarantine and Plant Health Officer at the RBGE, and she's studying the ecology of pathogens in traded plants. And her talk today is about biosecurity, biodiversity, and the pandemic. So over to you, Katy.
I'll just jump right in by wishing everyone a happy International Year of Plant Health. Certain other issues have overshadowed this, what was to be a special year for that reason. But in this talk, I'll be going through a little bit of how some of the concepts that have unfortunately become all too familiar to us this year apply just as well to plant diseases. I'll plug two special issues that I highly recommend you check out. This year's Sabaldia from RBGE and the BG Journal from BGCI are both dedicated to plant health and biosecurity and conservation horticulture, and both are available freely online. Today, this talk certainly won't be the first time that you've heard that emerging plant diseases threaten biodiversity. The IUCN reported last year that more than half of Europe's endemic trees are threatened with extinction, and a huge part of that threat is from invasive and emerging pests, including pathogens. It's difficult to miss the harm that emerging diseases are doing to Scottish plants. You may have seen juniper, common juniper, going bronze and then brown and then skeletal, like these in Glentrool. They were infected by Phytophthora ostracedri, which is the same pathogen that killed the iconic Juniperus rigida in RBGE's rock garden. And if you're in Edinburgh, you certainly don't have to go very far to find emerging diseases. The cycle paths are lined with dying ash trees, and I expect it's probably the same in many UK cities and towns. And it's not to mention the elms that have held on for so long and are slowly going down to Dutch elm disease. And you may even have seen these golden root mealybugs in your home garden. They're a recent introduction from New Zealand. Unlike most mealybugs, they can overwinter outdoors in Scotland. They're hard to see and really difficult to get rid of. These are they. You might think they were just a speck of vermiculite. But there they are, and they've become widespread in the horticultural plant trade. They have a wide host range, and most plants can tolerate some level of infestation, but some, including Primula and Meconopsis is one of importance in ornamentals, struggle with even small infestations. So while we know that ash dieback and ostracedri are really harmful for our Scottish biodiversity, we still don't know what will be the ultimate outcome of this invasion. And to be clear, I'm not saying that this will cause the same kind of widespread disease, but we just don't know. But I do suggest that you check the root zone of any plants that you buy. Now, this year also marks the end of the UN Decade for Biodiversity. The Coalition on Biological Diversity released its final report last month, and sadly, we did not meet those targets set out 10 years ago. Now, if business continues as usual, the CBD projects that we'll continue to lose natural habitats and forests, along with more conflict with animals and more risks of contracting new diseases from them, like the virus that has changed our lives this year. And we're on track for 3 degrees warming by the end of this century. Now, the UN has declared the next decade, starting in 2021, to be the decade on ecosystem restoration. And the CBD has laid out a set of 20-year targets for interlinked and interdependent strategies to move towards sustainable and comprehensive biological diversity conservation. Especially of interest to plant health and conservation are transition strategies toward coordinated land and forest management, as well as climate action, sustainable agriculture, and food. But most of all is the One Health transition that they've highlighted as an overarching framework and is a need to understand the links between conserving genetic diversity and individual species, community, and ecosystem health. This broad view is very much needed. We know that most invasive forest pests in the US and Europe were imported on plants for planting. And we've seen that high traffic sites, or especially those that are close to plant trading operations or to large scale plantings, whether they're commercial or for conservation, are at risk for new pest introductions that can lead to emerging disease. But risk factors for new introductions are not the only issue. The bark beetle outbreaks in the US and Europe in recent years, as in this photo, are a good illustration. The beetles, as I'm sure this is a familiar story, the beetles are attracted to stressed trees. Hotter, drier summers are giving them those stressed trees. And it's been compounded, especially in the US, by a shift from indigenous management to fire suppression. And dense stands with a lot of dead wood from beetles and from other diseases, like Phytophthora ramorum, in fact, have helped to contribute to recent years' catastrophic fires. And these have often burned hot, gone up into the upper canopy, killed the mature trees that would or might normally benefit from a burn. And speaking of Phytophthora ramorum, the sudden larch death epidemic is an example of these kinds of interacting effects. Phytophthora ramorum emerged in the UK in 2000, I have 2005, I think it's 2004, in parks and gardens. It was introduced through the plant trade on ornamentals, where it causes a foliar blight or can also be completely unnoticed. It during this early epidemic would cause sometimes fatal infections on hardwoods, but that wasn't the primary epidemic. In 2009, it made a host jump to larch, and you'll have seen the hillsides of larch plantations either dead or being cleared. Larch produce more spores when they're infected than any other host. They can do this before they show any symptoms. That is, they can be pre-symptomatic super spreaders. In fact, combined with the fact that they're grown in dense hectare scale stands with no social distancing, this creates an epidemiological nightmare. We know that a number of Scottish native plants are susceptible to ramorum, quite a large number. And we know that a number of hosts are moderately susceptible. That is, they can get infected, but only if there's a high spore pressure, like if they're growing under a larch tree. We also know that degrees of susceptibility can be overcome by evolution on the pathogen's part. And whether that's likely to happen or not depends on quite a lot of different factors. But the high spore pressure alone creates a new risk to Scottish plants that we might do better to avoid. This brings us to a central paradox in conservation and restoration horticulture. Live plant movements are the primary pathway for pest introductions. Growing and translocating plants is essential for conservation and restoration. So we in conservation horticulture have to reckon with how to reduce risks associated with propagating plants and with conservation restorations, in fact. But this is one of the biggest issues is in nursery propagation. And that's because being a pathogen and invading as a pathogen can be rather difficult. Pest introductions are a numbers game. Of all the propagules that land, very few will probably find a susceptible host and take off. So I'm talking about landing from a distant area into a naive population. But conditions that are good for growing plants are also good for growing pathogens. And we've seen this year how exponential curves work. The larger an outbreak, the more difficult it is to control. And when populations build up, there's a greater chance of adapting to a new environment or to new hosts. And regardless, larger populations are less likely to die out by chance, and more likely to be transmitted to other nurseries, to other conservation sites, or out into the wider environment. I don't intend this to be a comprehensive instruction on biosecurity. If you're interested in more details, the special issues I mentioned are worth following up on. Also, there was a great session in the Sabaldia 350 and Plant Network Conference this past week with Suzanne Sharrock of the International Plant Sentinel Network and BGCI. I'll just highlight that just as in emerging human diseases, we see central themes of controlling movement, of inspection, isolation, and quarantine, of keeping hands and surfaces clean, feet, tools, equipment, everything else, and even contact tracing when serious diseases are found. I'll also highlight that I've mentioned here monitoring with different methods for cryptic pathogens. So using molecular detection, isolation by culture. In the seed session, X-rays were mentioned as well. And this is to detect pathogens that may not be causing symptoms, but that are transmitted, that we know are transmitted very easily in nurseries. Phytophthora ostracedri is one of these, and it's believed to have been transmitted to glandrule and other sensitive sites through juniper trees for restoration projects. Liam Morton won the BSS dissertation prize this year for his undergraduate, his honors thesis showing that Phytophthora ostracedri is causing severe disease on Xanthocyparis vietnamensis, which is an endangered conifer in ornamental use and in collection at the botanics. Now, the last thing we want to do is to inadvertently transmit a pathogen like Phytophthora ostracedri to conservation nurseries in Vietnam and then back into sensitive sites, because in these highly restricted ranges, the plants just can't take another stressor. But it's not always clear what these findings mean, particularly with molecular findings. There are a lot of pests out there. The more we look with newer tools, the more we learn about them. On the left is a figure plotting all the known Phytophthora species as of last autumn by the year they were first described on the x-axis. On the y-axis is the number of host genera reported on the USDA host pathogen, actually host fungal, but oomycete, host fungal-like database. So this is the number of host genera. This is the year the Phytophthora species was described. I've put a dividing line at 1991, which is the year that God and Bruns published ITS timers as a DNA barcode for identifying fungal species. It also works for oomycetes. Before that line, there are 42 species described. After that, 119. And we can see as well, it's remarkable how the number to the right is quite a lot lower than, well, at least up to 1930. It is interesting that between 30 and 91, only specialists were described. I'm not sure what to make of that, but it is fascinating. But here we do see that quite a lot of these have been reported on just a single host or from water or a substrate without any real disease symptoms. But we do see there Phytophthora morum and Phytophthora ostracedri. Now, the figure on the right shows us something else. It takes all the species described in before 1996 here. So this is host genera. So this is all of these species, the dots. Each dot is a species that was described prior to 1996. On the x-axis is the number of host genera when they were described or when they were treated in Irwin and Ribeiro's very comprehensive Phytophthora Diseases Worldwide book. This was the source. On the y-axis is the number of host genera in the database in 2019. Now, as we'd expect, there is a bit of an increase. The intercept is at 4, and the slope is a little bit more than 1.5. So the intercept at 4 tells us that specialists had a few more hosts reported in 2019 than 96, as we expect, because knowledge advances, but they largely remained specialists. However, the more generalist the species, the more hosts it gained. And just for reference, this is Phytophthora citricola. In 96, it had 49 host genera. It's now 23 years later at 116. So we wonder a bit, although it's a bit of a maybe apples and oranges, but we wonder if in 23 years, if we won't be up to 100 or more hosts for Phytophthora ramorum. And this is important. It's not just academic, because while European plant health laws are shifting to a more general approach, statutory decisions on both formal and informal risk assessments are nearly always made on the basis of known host relationships. So we need to have a way of knowing the difference between a species with 1 or 2 hosts that we haven't seen much disease from, that's going to stay that way, one that will become a serious pathogen, whether of a few or many hosts. So to sum up, climate stress, species invasions by global trade, and land use change are synergistic threats to plant biodiversity, and as such, they need a multi-pronged and coordinated response. Restoration and climate mitigation works for biosecurity, conservation, and resilience are necessary and important, and I'm not arguing for a minute they should stop, but they can create added risks of pest introduction. So biosecurity and conservation is essential to mitigate these. And the challenge remains to better understand new and unknown threats and to achieve resilience to the unknown. Thanks very much to my funders and my colleagues, and thanks very much to you for your attention.